Humans have a persistent psychological drive to organize the natural world, historically grouping organisms by simple visual similarities and shared behaviors, the lines between predator, prey, plant, and fungus. In 1753, Carolus Linnaeus attempted to force a mathematical order onto this biological chaos with the publication of his text Species Plantarum. He introduced binomial nomenclature, a system that assigns every known organism a two-part Latin identifier, a generic name indicating the genus, and a specific epithet. Over centuries, that two-term system ballooned into a tiered architecture. Naturalists grouped organisms into broad categories based purely on shared physical traits, like feather structure. Taxonomists created absolute ranks to contain them, moving top-down from kingdom to phylum, class, order, family, genus, and finally species. You can see how this works by tracing a highly recognizable animal. The red fox starts at the specific epithet vulpes. It sits inside the genus Vulpes, which belongs to the family Canidae. That family rolls up into the order Carnivora, and finally the class Mammalia. The structural flaw here is historical. Linnaeus designed this rigid hierarchy in a fixist era, roughly a century before the discovery of evolution. Evolutionary branching is fluid and continuous over deep time. There is no objective rule dictating how much variation constitutes an order versus a family. This diagram illustrates the boundary paradox. You cannot neatly overlay an inverted pyramid of discrete, human-invented ranks onto a sprawling, continuous, time-scaled phylogenetic tree. The rigid tiers simply fail to align with the organic branching nodes of actual evolutionary history. Because rank-based nomenclature conflicts with the continuous reality of evolutionary data, taxonomy requires a transition to computational classification to remain accurate. By the mid-20th century, taxonomic progress stalled. Scientists were still trying to map deep evolutionary relationships entirely by measuring physical phenotypes. That morphological approach breaks down entirely when classifying microorganisms. Bacteria and archaea lack the physical characteristics to visually establish their relationships. To bypass the limits of human observation, researchers turned to molecular systematics, using genetic sequences as an objective ledger of evolutionary history. In 1990, microbiologist Carl Woese mapped the deepest branches of life by comparing ribosomal RNA sequences. This diagram represents Woese's 1990 phylogenetic tree, mapping the divergence of living organisms into three primary branches from a single root. The RNA data revealed a structural revelation. Archaea and eukarya share a more recent common ancestor with each other than either does with bacteria. Bacteria and archaea look nearly identical under a microscope, but their DNA reveals massive evolutionary divergence. This evidence dismantled the old kingdom models. Woese established a new, higher taxon, the domain, splitting life into bacteria, archaea, and eukarya. Deep evolutionary relationships can only be accurately tracked through molecular genetics. Purely visual, rank-based classification fails at the margins of life. While geneticists were successfully mapping molecular trees, the administrative side of taxonomy lagged decades behind. The formal naming of organisms was still bound by 19th century rulebooks. Two primary legacy codes governed the field: the International Code of Zoological Nomenclature for animals and a separate botanical code for plants and fungi. These codes enforce highly subjective rules. They mandate absolute ranks and standardized Latin suffixes to designate families, locking organisms into rigid taxonomic boxes. This bureaucratic system created a structural mess, producing intercode homonyms, where the exact same name was applied to different organisms across codes. Attempts to unify them under a single biocode collapsed due to entrenched academic traditions. The solution arrived in 2020 with the Phylocode, an objective alternative to the legacy systems. Phylocode entirely eliminates the requirement for absolute taxonomic ranks. There is no mandatory class or order. This compares the approaches. On the left, ICZN stacks organisms in rigid tiers. On the right, Phylocode relies on clade definitions determined by branching nodes. These highlighted specifiers anchor the name to a specific point of evolutionary divergence instead of an arbitrary tier. By abandoning mandatory ranks for node-based definitions, science established a nomenclature system that bends to fit evolutionary history, rather than forcing history to fit nomenclature. Phylocode and ribosomal RNA solve the problem of deep evolutionary mapping. But out in the field, identifying a physical specimen still requires observable morphology. This creates a major operational bottleneck with cryptic species complexes, organisms that have diverged genetically but still appear visually identical to the human eye. Look at these patterned conus shells. Differentiating them relies on analyzing fine-grained visual geometries that often overlap between distinct genetic lineages. Traditionally, taxonomists must independently decide which traits, like shell curvature, rib patterns, or color metrics, are diagnostically important. This introduces heavy human bias into the measurement. To strip subjectivity from morphological measurement, researchers are deploying deep convolutional neural networks, or CNNs. A CNN performs autonomous feature extraction. It learns to recognize relevant visual cues directly from raw image data, without relying on predefined human labels. As an image passes through the hidden layers of this CNN architecture, the network isolates complex structural geometry. The output is visualized in a dimensionality reduction scatterplot, organically clustering data points to separate identical-looking specimens based purely on rigorous pattern recognition. CNNs eliminate human observation bias, converting morphology from a qualitative art into a mathematically reproducible science. Relying on a single data stream is insufficient. CNNs cannot resolve genetically identical color morphs, and pure DNA sequencing ignores the ecological context of a living organism. Modern species delimitation requires integrative taxonomy, the simultaneous, algorithmic analysis of multiple disparate data sources. To process this highly complex, multimodal data without prior human labels, researchers use unsupervised machine learning, specifically self-organizing maps, or SOMs. This visualizes a self-organizing map architecture. 3 distinct data streams, morphological shell clusters, genetic RNA barcodes, and spatial ecological coordinates feed simultaneously into a central node. The node calculates a high-confidence boundary, reconciling these physical, genetic, and geographic variables into a definitive, rankless species cluster. These algorithmic tools do not render the human expert obsolete. They eliminate the tedious, biased sorting that historically bogged down classical taxonomy. These tools allow taxonomists to delegate sorting and identification to algorithms, focusing instead on the deeper synthesis of evolutionary relationships. By replacing 18th-century ranks with multimodal algorithms, biological classification is becoming a precise, data-driven reflection of the history of life on Earth.